Hello everyone, my name is Medea Raza, and today I'm honored to present on rebuilding hearts with CRISPR-Cas9. I'm a rising senior at Alliance Academy for Innovation in Cumming, Georgia, and I'm deeply immersed in science, research, and global health. Through my nonprofit called Heart to Heart with Medea, I've shared cardiovascular awareness with over 159,000 blog readers and 70,000 YouTube viewers. I've authored a book and traveled through 10 countries collecting research and stories about heart health. My lab experience includes investigations into EMS displays in rural Georgia, CRISPR modeling of cardiac arrhythmias, and hands-on cardio-oncology research at Grady Hospital as well as Emory and Augusta University under my mentors Dr. Guha, Dr. Wemberly, and Dr. Smith. Today, I'm excited to explore how CRISPR can transform cardiovascular disease prevention and rebuild our biological hearts from the inside out. Cardiovascular disease is the leading cause of death globally, claiming over 17.9 million lives each year. That's roughly 1 in every 3 deaths. Most of these deaths are preventable. In many parts of the world, even access to a defibrillator or a beta blocker is limited. And even in countries like the US, we wait until someone's heart fails before treating them. That model is reactive, not proactive. But we know that genetics plays a massive role in cardiovascular disease, both in triggering it and accelerating its impact. If we could intervene at the genetic level, early on, we wouldn't just be adding years to life. We'd be giving people more stronger, more resilient hearts from the start. That's where CRISPR comes in. CRISPR-Cas9 technology acts like molecular scissors, guided by a custom RNA sequence to locate a specific DNA target. Once the Cas9 enzyme cuts both strands of DNA, the cell triggers its natural repair pathways. We can exploit this mechanism in 2 key ways. If we allow non-homologous end joining, we can disrupt faulty genes. Or, by providing a DNA template, the cell can repair itself through homology-directed repair, allowing us to precisely correct disease-causing mutations. This unparalleled precision makes CRISPR a powerful tool in genetically targeting cardiovascular diseases at their very root. Let me walk you through a potential therapeutic pipeline. First, we extract induced pluripotent stem cells, or iPSCs, from a patient. Next, we use CRISPR to correct or knock out a pathogenic gene, such as MYBPC3, which is a common culprit in hypertrophic cardiomyopathy. The edited cells are differentiated in the lab into functioning cardiomyocytes. After validating that the cells exhibit healthy behavior in vitro, these cells can either be reintroduced directly into the patient, or we can take an alternative approach by delivering CRISPR machinery directly to the patient's heart in vivo. This method has shown success in preclinical models, where edited cells reduce cardiac fibrosis and normalize heart muscle structure. Hypertrophic cardiomyopathy affects approximately 14 million people globally. Many are asymptomatic until they suffer sudden cardiac events, often in adolescence or early adulthood. It frequently stems from mutations in sarcomere genes like MYBPC3 or MYH7. At the University of Pennsylvania, researchers are pioneering base editing techniques and somatic CRISPR therapies to precisely correct these mutations in animal models. But the scope goes beyond HCM. CRISPR is applying to monogenic arrhythmias, hypertensive diseases, and even lipid disorders. For example, editing genes such as PCKS3 or ANGPTL3, which are known to regulate cholesterol and could form the basis of one-time genetic prophylaxis, essentially a cardiovascular vaccine. Efficient delivery of CRISPR components remains a critical challenge. Adeno-associated viruses like AAV9 are optimal for targeting cardiomyocytes. Another promising method is lipid nanoparticles, combined with ultrasound-enhanced delivery. Recent advances in mouse models have demonstrated editing efficiency of 30 to 60% in heart tissue, with significant functional benefits. Even more cutting edge are split adenine bases that activate only in the presence of a trigger molecule, reducing off-target effects and enhancing safety, which are key improvements as we move from animal models to human therapies. Looking ahead, the future of heart health lies in personalized and predictive genomics. Polygenic risk scores enable us to identify individuals at heightened genetic risk for heart disease years before symptoms emerge. Combining these risk assessments with CRISPR interventions means we can potentially prevent disease before it starts. Picture a future where newborns undergo genetic screening and receive precise gene edits to correct pathogenic variants, drastically reducing lifetime cardiovascular risk. These one-time interventions could dramatically shift public health, especially in underserved regions where ongoing treatment is less accessible. We are at a scientific inflection point. CRISPR-Cas9 enables us to reimagine cardiovascular care as proactive, preventive, and genetic. I envision a world where heart failure becomes a relic of the past thanks to targeted genetic intervention. But achieving this future demands rigorous science, ethical stewardship, and international collaboration. So I ask you, when heart disease is a question of choice, will we choose to prevent it? I've included critical references from the World Health Organization, recent cardiovascular gene editing literature, Penn Medicine, and NIH resources. Thank you to the Global Health Leaders Conference at Johns Hopkins University for the platform to present my research. Thank you for your attention today. I welcome your questions and ideas. Feel free to contact me. Together, let's engineer a future where heart disease is preventable and global health is redefined. Thank you.